because this is a black cat with a very, very obvious white locket and some even, the white's very diffuse. It's even going away from the locket. I didn't want to pick an Abbey or a Somali picture. I thought we'd just go for a neutral moggy because it is a fault on our breeds. And that is something I'm going to talk about when we get to white. But we've got a bit of a way to go before then. I'm going to start with a very brief whistle stop talk of cats in general, then our breeds, and then we'll go into some general genetic terms, most of which you don't need to learn. So if you're not quite there yet, don't worry. I'll tell you what you need to know, what you need to understand to go further. We'll then do color, coat length, and patterns primarily that are related to us. I'm not doing all of the color point and different things that we don't need to know for our breeds, but there are some things that you do need to know, even if they're not in Abyssinians and Somalis, because um, it's part of the genetic makeup of our cat that you understand why you get that and why you get this. There will be a little bit of that, but not much. And I've just put this little picture here for interest because we will be talking about the red series and torties and things later. And that's just a nice, easy little graph. And that is basically about as complicated as we're going to get tonight. Oh, species of cats. So cats have been around as long as there's been mammals. We have three subsets of cats. The large, which would have had the cyber tooths and things that are now extinct. We've got all the big cats that are alive on the planet now. You've then got the medium group, the lynx, pumas, ocelots, type things. And of course, the Asian leopard and things that people have outcrossed too for hybrid manufactured breeds. But most domesticated cats will only have the small cat genes, and these are the wild cats that evolved in Africa and the Middle East. I'm just going to have a little sip of my tea, just my mouth gets a bit dry when I'm talking. You'll hear the occasional slurs. So we know that wild cats were domesticated about 10,000 years ago. It was sort of towards the end of the Stone Age and the Neolithical period. Agriculture seems to have gone hand in hand in that. When we were hunter-gatherers living in groups of 10 or 20, there wasn't a huge need to store food. Obviously, food would have to be stored for winter, but not in massive quantities because we were small groups. By the time agriculture really got established, which started mostly on the banks of the Nile because it was just such fertile growing area because of the Nile and the nutrients it had brought to the surrounding soils. Suddenly, we had towns and cities of thousands, and the need to store food grew exponentially, which attracted massive numbers of rodents, which would obviously eat the grain and contaminate grain that they didn't eat and bring disease and problems. So the cats probably turned up in a response to the rodents. They probably went, oh, there's a new food source. The grain and the vegetables attracted the rodents, and the rodents attracted the cats. And we very quickly established a symbiotic relationship with the cats. The Egyptians, of course, famously elevating them to godlike status. But humanity would not be where it is today if we hadn't had cats at this early stage. It was before we could have coped with other methods the food sources would have been diseased or lost, and we would have died out, or certainly not have got to the stage that we've got now. We wouldn't have been as successful. And this is really well demonstrated in lots of archaeology findings throughout the fossil record, that at the birth of agriculture, we suddenly start to find cats living with us and amongst us. And one of the easiest ways to explain that is until 10,000 years ago, there was no people on the island of Cyprus, and there was no cats either. There was all manner of other creatures and birds, but there was no cats and no humans. And as soon as humans arrive, as soon as there's evidence of fire, human bones and things, there is, cat poo and cat bones at exactly the same time in the whatever archaeologists dig in the archaeology findings. So we know that they didn't have advanced boats in those days. They only had little wooden rowing craft, but still they must have brought cats with them right from the get-go. A lot of us breeders very much like the idea that the Abyssinians were, of course, the hieroglyph cats of ancient Egypt, because it's 4,000 years ago. We don't have any written record to support it, but there is a likeness to Abyssinians and the hieroglyphs, and we do know with fairly good evidence that certainly the first Abyssinian came from what is now known as Ethiopia, Abyssinia. She was exhibited at the first Crystal Palace show in 1871, and there was a follow-up article in the newspaper in January 72 where a picture of the cat was printed with the story. It was the wife of a captain returning from the war there, and he brought a female kitten back with him called Zula. She's described as a ticked tabby, likely with quite a lot of leg barring, possibly some tail rings still, but essentially a ticked tabby cat. And remember, photographs were very much in their infancy around this time. They were only black and white. They weren't of the high definition that we have now. But there was a picture of Zula in the book published in 1874, Cats, Their Points and Classification, where Mrs. Captain Barrett Leonard again was interviewed and gives her take on the story of Zula coming in. She was described as having a moderate coat length at the 1871 show. This picture depicts a short-haired cat. The picture that was drawn at the show, she did look longer. And it's believed that there was probably always long hairs. They were probably very rare because there was only a few carriers, but they were probably always present, as they were in the British short hairs and the Russian blues. But certainly long-haired carriers were more common after the Second World War. There was only 12 Abyssinians left in 1847 when the club reformed after the war, and other cats had to be used as outcrosses, including a Russian blue, who was widely used, I think, at the time. But Rebecca will know a lot more of that history than me. This was just a little bit to give you a snapshot as we go forward. Is that kind of speed that I'm talking about all right with anybody? Because that was just a little whistle stop. Got a couple of those up there. Awesome. So general genetics. All cats are black and all cats are tabby. And I'll continue to explain that as we go through. Obviously, these early wildcats would have been black tabbies. Everything else that we have now is a mutation. And most of it came about as a natural mutation, and it succeeded because it either worked very well in that environment or at least it didn't hinder. So some things will work better in different environments. If you live in the desert, you need to be able to adapt to the very hot daytimes and the very cold night times and the shortage of water and that kind of thing. So things that would help you where you are would flourish very well. And if it didn't hinder, then it wouldn't die out. But obviously, if the other cats had an edge over a cat with a certain feature, then the one with the edge is going to succeed. It's going to produce more kittens. It's going to live more successfully, and it's going to pass on that feature much more. Cats have 38 chromosomes, 18 pairs, which they inherit one from each parent, and the two sex links, the X and the Y. And they have approximately 20,000 genes, which is similar to us humans. The number of chromosomes you have doesn't demonstrate any degree of complexity or intelligence, because just as an example, we have 46, but dogs have 78, and there's a type of hamster that actually has 92 chromosomes. So there's no relevance really over how advanced you are and the number of chromosomes that you have. So all mammals get a complete set from each parent. The cat obviously gets 18 and the X chromosome from its mother, and 18 and the X if it's a female kitten from a sire, or 18 and the Y if it is a male kitten. And that just helps give us a little bit of a starting point. We are going to talk about probably less than 15 or 20 genes. And I am primarily just going to use the term gene. There is a difference between genes and alleles, but at the level that we're looking at, it doesn't matter. I don't need to differentiate. I keep it straightforward. So if I use either term, I'm using them interchangeably at this level. Don't worry. Now, these are some of the big words that you don't need to remember. Dominant and recessive is something that I'll need you to understand to go forward, but the rest or not. We can talk about them in easier common terms, but I will just touch on them briefly now. I'll have another sip of tea. Genotype. So that is the genes that the cat has. If you do a swab and you send it off and it comes back and it tells you exactly what genes the cats have, that's the genotype. The phenotype is what it looks like. So the cat doesn't necessarily have to look exactly like its genes are. A usual cat can carry dilute and throw blue kittens, so its genotype will reflect that it's a black cat and that it carries dilute. But you can't see that it carries dilute by looking at it. You just see the usual or ruddy Abyssinian. You don't see looking at it that it carries blue. That's in the genotype, but it's not expressed in the phenotype. And I will use a few examples as we go through where I talk about that, but it's essentially just because it's got a gene doesn't mean it's expressed, doesn't mean that it shows. Homozygous are where two are the same, and heterozygous are where two are different. So there'll be lots of examples of that for all the colors and patterns as we go through. If they get the same gene from both parents, they're then homozygous, they've got two the same, they can only pass that one on to their offspring, there's no other option. If they get a different one from each parent, then it's a roll of the dice, which one they pass. I'll often say it's genetically, it's 50-50. But if you've been breeding a long time, you know that it's never actually 50-50 in practice. You can roll 6 ones off the bat quite easily. Anything that is possible will definitely happen if you give it long enough and enough opportunities to express itself. It will come. But essentially, all things being equal, it should be 50-50. So these different genes that we all have, some of them are dominant over all other genes. Some of them are recessive over all other genes. Some of them are in between. So they're dominant over some genes, but recessive to others. And that's where you get some of the more randomness of things that can pop up. There is also an incomplete dominant, and we do have 1 or 2 examples of that later on as we get into the colors. But it's not common, it's not the norm. For the vast majority of time, a gene will be dominant and or recessive, and it'll be that straightforward. There won't be, but what if? It's more rare. There are some polygenetic traits. Most of these are either known or suspected, but not yet fully mapped. And examples of these in cats are size and eye color. So you can mate 2 small cats together and potentially get some quite large offspring, and it's because there's more than one gene that governs that. And because you only know what your cat looks like, or perhaps the parents, the pedigree wouldn't really reflect size very well, unless you're very familiar with the lines, you can get some surprises with the size of cats and eye color and clarity of eye color as well. All colors are either black-based or red-based, and the red cat will still have the black-based colors, but the red overstamps. So the red is carried on the X chromosome only, but it will overstamp any primary color that's already there. Would anybody like to ask questions at this stage? Okay, I'm going to drink my tea and then we're going to move on. Primary coat colors. So all of our small cats, all domesticated cats, have 3 primary coat colors. Black, which in our breeds is usual or ruddy tick tabby, chocolate, and cinnamon. Genetically, it's cinnamon. In Abys and Somalis, we call it sorrel, or, which is cinnamon tick tabby, but that is because the name for the color was given before we genetically knew that it was cinnamon. It's not red, it's not the sex-linked red, it's a different color altogether. And that is the order of dominance. Black, then chocolate, then cinnamon. So, and these little letters on the side, they denote the B gene, which is the black gene, and how that's expressed. So the capital B is the black, the usual cat, and it can have 2 copies the same. It can be homozygous, so it can be a usual cat. It doesn't carry any other primary colors. it will only pass on the usual gene to its kitten. It can be a black cat that carries chocolate. So when you get your swab results back from Langford's or whoever you might use, you will see that it's capital B, little b. Or potentially it can be usual carrying sorrel, the cinnamon gene. And then if you have a chocolate cat, it can be homozygous chocolate, or it could be a chocolate cat carrying sorrel. Your sorrel cat only has the two cinnamon genes. It doesn't have another primary color. It cannot carry usual or chocolate. Sorrel is the bottom run of the ladder. If a cat is sorrel, it doesn't carry any other colors. But the chocolate can carry cinnamon because it sits in the middle. It's recessive to black, but it is dominant to cinnamon. So it sits in the middle. But a usual cat can potentially carry either of the other two colors. And sometimes you know this because of the pedigree of the cat. And sometimes you can't guess because it just depends. You don't know what the other cats were carrying. So you know what color the parents were, you know what color the grandparents were, but you don't know if they carried anything. And before we had genetic tests, breeders had to do test matings if they wanted to know, if they didn't want to get any surprises, or they were looking for a particular color or making a color more prominent in the breed, they would have to do test matings to see what they get. So they would go to a cat that they knew carried a certain color and see if they got any. And of course, it's a roll of the dice, so you won't necessarily get some in each litter, but that was the only way to do it. We're very lucky now that we know. Well, there's your three basic colors. So every single one of our breed, their B group color gene will be one of these ones I have listed on the left of the screen. Doesn't matter what color it is, it could be a torty, it could be anything. It will be this at the B locus. This is what it will have, one of these. And now I'm going to go to the dilute colors. So our usual chocolate and cinnamon sorrel cat, they are not dilute. They could carry dilute or not. If they don't carry dilute, they'll be big D, big D. Don't carry dilute, won't ever produce any dilute kittens. Both parents have to carry or be dilute to get dilute kittens. They could be big D, little d, where they carry dilute. It's not expressed on them, but they carry it. So your usual queen that sometimes has some blue kittens, she has to be big D, little d. And if she is in fact dilute herself, she's a blue cat, she'll be little d, little d. So she'll be black, black, but then little d, making the black blue. And it's actually just, it is a dilution. So the cat is still black, but it's expressed as this gray color because the dilute bleaches some of the color out. And the chocolate cat is expressed as lilac when it has two copies of the dilute gene, and the cinnamon sorrel cat is expressed as fawn. So at this point, I'm going to introduce Tabitha, and she's just a cat that I'm going to make reference to as we go through the slides. She is a fawn silver Abyssinian. So at the bottom there, you can see she's B1B1. That is the cinnamon gene, two copies the same. She's homozygous cinnamon, but because she's also homozygous dilute, it's expressed as fawn. So far, we haven't got to silver yet. We just know that she's B1B1 and little d, little d. And that's it. Any questions about dilute carriers, dilute kittens? Please don't be shy.
Yeah,
If you
I've got a question, Nathan. Sorry, can I just go over the expression of the dilute? Could Tabitha, who's B1,
yes,
little d, and you said she was fawn, could she have been oh God, I'm gonna get mixed up now. Fawn is the dilute of sorrel, is that right?
yes,
Yeah.
yes.
So could she have looked sorrel but still be a little d, little d? It's just that the sorrel was expressed. So she was dilute, she's got dilute, or would
Yeah.
it always be capital D/D?
Yes, she will always be that. If she is a fawn, that will always be a genetic makeup. You can get some variance on the degree of dilute expression.
Yeah.
So, like you can get a hot red or a hot cream, you can get a dark blue or dark fawn or lilac to some degree. And I've seen fawns where you go, is that really fawn? That's quite a dark fawn. So
Okay.
yes, you can get
Thank you.
degrees of expression, but the swab results will always come back the same if that's what she is.
Cool.
And also, by all means, if anybody wants to make a note of any questions that they have at any stage and they want to wait till the end when we stop the recording and ask the questions, then that is absolutely fine as well. And not everybody wants to be recorded for posterity. And people might think that that question's stupid, but really there are no stupid questions. And I just want to check that I'm not talking too fast and I'm covering anything. So tell me to slow down or go over something again or explain it in a different way if you would like. So that is the dilute colors. So I'll just go back. We've got the three primary colors there: black, chocolate, and cinnamon sorrel in the order of dominance. So then, therefore, in your dilute cats, the order of dominance is blue, lilac, depending on what genes it has at the B locust, and if it has the two little d's at the D locust. So then, just because some of you crazy people like silvers, I've included the silver inhibitor gene. So this is really quite an interesting gene for a lot of reasons. So the first thing is a cat doesn't actually have a silver gene. It has an inhibitor gene and it restricts the expression of the other colors. So it gives pale gray light bands, particularly at the base of the hair. But it's bleaching the color that's there. So the cat is still usual chocolate sorrel and or dilute. But the color isn't expressed as that. You've got this pale gray, which looks white at the base, because it's been bleached away by the silver inhibitor gene. And it's the gene that we have recently mapped and is now available for swab testing is a dominant gene. There may be some degree of incomplete dominance over things, and there might also, well, we're pretty sure there are also other lesser genes that can influence or make a cat be silver. But this is the main culprit for silver, is the gene that we've mapped, the I gene. It's a dominant gene, so the cat only has to have one copy to appear silver, and you can't tell by looking at the cat whether it's got one copy or two copies. You only know that it either has at least one or it doesn't have one. And sometimes when you get I'm talking widely across breeds now, not just ours, you get cats that you think is this cat silver, but it doesn't have a silver parent, but it's the blue tabby's not just blue, it's there's a silvery sheen to it. It's got a much stronger white undercoat than it should have. Could it be silver? And this test may still come back negative because there are still some genes that we haven't mapped yet, that we don't fully understand. We just know this is the main culprit for silver, but it isn't the only one. And there might be some polygene influence as well. So certain colors and patterns will be more inhibited and show the silver more than others. And we'll get to that when we talk about tabbies as well. So just going back to Tabitha, so we don't know if she has 1 copy or 2 copies of the silver. We would only know that if we swabbed her. Or I suppose potentially on her pedigree, but not necessarily. But we know that she's B1B1. We've got 2 copies of the fawn gene. 2 copies of the sorrel gene, and she's little d, making the sorrel be expressed as fawn, and then we know she has at least one copy of silver. And I suppose we would know that she only has one if she only had 1 silver parent. If she had 2 silver parents, then she could potentially have 2. She could have got 1 for each parent. But the non-silver parent could only give her the non-silver gene. So, yeah, she could have 1 or 2 copies. And we'll come back to silver. So I want to talk a little bit. No, I'll talk about it now. So, because it bleaches the base of the hair, it gives you that white appearance at the base. When you mate silver to silver, that can creep up the hair. That could come further up. And we know this because the chinchillas, the silver Persians, mate chinchilla to chinchilla, and after however many generations, it's not many, but say somewhere between 2 and 4, they start to almost lose the black tipping. They don't have ticking, they have a black tip on the end of what appears to be white hair. So they have to outcross every so many generations. They have to go out to another color and then breed back so that they don't lose it, because eventually they would end up with an almost white cat that wouldn't genetically be white, but they would have so much of the silver inhibitor that they would lose the color even on the tip of the head. An unacceptable amount of white and black. So that's something to think about if you breed silvers, especially if you have silvers that are carrying 2 copies of the silver gene. If you mate them together, you've got nothing else in the mix. And you might see sort of white creeping up, often from the extremities and the tail, but potentially over the body to some degree. So then somebody thought it would be a really good idea to introduce the sex-linked red into the Abbeys and sort of then secondary by proxy into the Somalis. So this is really well known from all the other breeds. It was probably even before the gene was mapped. It was understood because of the 100 years of organized cat breeding and registering. We really understood how the red series worked. It's carried only on the X chromosome. Therefore, males who are XY, they cannot have more than 1 copy. They only have 1. Females can have 2 copies potentially of the red gene if they get 1 from each parent because they are XX. And when they only have the red on 1 of the chromosomes, on 1 of the X chromosomes, you can get tortoiseshell. And I said earlier, and I'll just recap now, the red overstamps the primary color. So the cat genetically still has the black chocolate cinnamon sorrel. It still has that, still has 2 copies of whichever combination it's got of that. It's still got that, but the red overstamps, the numismatic color overrides. And it does other things as well, but I'll get to that when we talk about tablets. So females can have 2 copies, and they will be then red or cream if they have 2 copies of the dilute gene. They can be big O, little o, and they'll be expressed as tortoiseshell. Obviously, like black tortoiseshell if they don't chocolate or cinnamon, and then if they dilute, one of the dilute colors, or non-red, so no copies of the red gene. Males only have the 1 copy, so they're either red or not red. Now, male torties do exist. They are very rare. I've included it in the slide just because at this point somebody normally always asks, what about male tortie? There's 3 ways you can get a male tortie. The true male torties are either chimeras, where the embryos were splitting, and it's basically a failed twin, and the male also has another set of genetic information as well as its own, and it's then able to express red and black, or I should say red and a primary color. Or the rare genetic syndrome where they're XXY and they're almost 100% sterile because it's genetic. They have an extra chromosome, and therefore they're able to be torties, but they're generally sterile. And I think like the 1 or 2 in 1,000 that aren't sterile are not particularly good breeders. Anyway, they have poorly developed testes, and they're not extremely fertile, and they're not extremely rampant. They're a bit asexual. So, when you, because I know there are some breeders, I don't think probably any who are on tonight, but some that work with the red series, and they're into the reds and creams and the torties. So I'll talk just a little bit about the interestingness of breeding. So if you are working with those colors and you like those colors and you are wanting to focus on those colors, then it's quite easy. Your red female will only have red offspring. She could have torties if you put it to a non-red male, but all of our female offspring will be red, no matter, all of our male offspring will be red. All of our female offspring will be red or torty, depending on the color of the male. So if you're working with reds and creams, you could quite exclusively or almost exclusively just produce reds and creams. It's such a strong overstamp of the primary colors. Your male red cats, all of their female offspring, they will pass the red gene to. So depending on the genes of the mother, the kittens will be either tortoiseshells from the male, or red if she also carries the red gene. And his male offspring will not get the red gene from him. So if you put a red male to a black female, you won't get any reds. You will only pass the red on to his female offspring. So Tabitha, go back to Tabby. She is B1, little d, and we know she's got at least one capital I for the silver, and we know that she's little zero, because she's not red. She's not a taut. She doesn't have. But if we introduce Mavis at this point, we don't know what she is at the B locus. She is a cream Abyssinian, a cream ticked Abyssinian. And it doesn't matter because the red has overstamped it. So yes, genetically she has something, but you will never get kittens in that color because she has two copies of the red gene, which will totally overstamp the black chocolate cinnamon. And if she's cream, she has to have two little Ds for the dilute to be expressed. And she's not silver, so she's little I. And that's the reds. Then I've included white, even though we don't have white in our breed. I just thought it was important, and I do want to talk a little bit about lockets and belly spots. So the only way really to do that was to give you a little bit of foundation in the white. So your white self cat has the dominant capital W gene. And it can have one or two copies. It's dominant. It only needs to have one copy to be expressed. If it was homozygous, capital W, all its offspring will be white, regardless of what color the other parent is. Obviously, the dominant white gene we know is associated with increased rates of deafness. So a homozygous white would have double chance of deafness. It would have two bites of the cherry. So breeders who breed whites will usually breed white to white because you're going to get increased risk of deafness there. And our cats will all be little w, because they're not white. We don't have the white gene in our breed. So they'll be whatever they are along the list, but then when we get to the W locust, they'll be little w. There is another gene that can give you white cats. It's a recessive white gene. It's very rare, and it's a true albino. And they have the pink. They don't have any pigment in the eye. The eye appears pink because they're true albinos. And they will genetically still have a primary color, etc., but it is bleached out because of the albino. Then there is the main white spotting gene, which has been mapped. It is dominant. It's expressed as capital W, little w. We don't have it in our breeds, but it is random. Whether they've got one or two copies, they can have quite a wide range of white. The white can be up to 100% when they have two copies, but even with one copy, it can be more than 50% white. So it's very difficult to control the white gene. I feel sorry for breeders of breeds with really strict patterns where they have the white spotting gene, because you might get one cat out of 10 kittens that show quality. And of course, the one that has the show pattern might not really have the type or the temperament for showing. So very difficult. I'm just going to have a little drink of tea. Then I wanted to move on to we haven't yet mapped it. We don't know. There may be more than one gene responsible for lockets and belly spots. Certainly one of the genes appears to be dominant because of what we've seen. So if you remember on the first slide, we had the black moggy with the big white locket in the middle. So that is not this white spotting gene. That is another gene, and they typically get the spot on the chest, and that's why we call it a locket, but it can be further down. It can be more on the underside of the cat. They can have more than one. And because there's no genetic test for it, you can't tell if your cats are carriers. We just know that it's dominant, so it would be best not to breed from an Abyssinian or a Somali with a locket or belly spot, because it will likely throw quite high numbers in its offspring, and then everybody ends up in a mess because it's a disqualifying feature at shows. Coat length, very straightforward. Short hair is dominant. And a variant will be big L, little l, and a short-haired cat not carrying long hair will be big L, big L. And your long-haired cat is just little l, little l. It cannot carry short hair. It is bottom run of the ladder. Short hair is the top run of the ladder. Long hair is the bottom run of the ladder. Variants should be expressed as short hair. They shouldn't have medium length hair. They should just have short hair, the same as their parents. The texture will be different. And we see that in the Abbeys and the short-haired Somalis. The short-haired Somalis have a very soft coat. It's not as resilient as the Abyssinians. The Abyssinians' coat is not soft, and it's got that lovely spring back quality when you brush it the wrong way. And the short-haired Somali, it doesn't have that resilience. It's much softer, and then that's just the texture of the hair has changed because it's a variant. But the length is dominant, so you shouldn't have a medium-length cat. It should still have the same hair length as a short-hair counterpart, but it carries potential to pass the long-haired gene on to the next generation. And when we talk about these genetic sort of recessive qualities, and especially something like this, it's a good example because you've only got long hair, short hair, nothing in between. You've got short hair dominant, long hair recessive. So you've got a cat that carries the carrier in the middle. It expresses the short hair, carries the long hair. What happens in the next generation? Well, it's a roll of the dice, 50-50. So it's going to pass the long hair gene on to half its offspring approximately, and the other to the other half. But in 6 generations, it could potentially still be there, or it could be lost after 1 generation, because each time you do the mating, it's a roll of the dice. Did they keep it? Did they pass it? Did they not pass it? Okay, they got it. They carry long hair or dilute or whatever, or cinnamon. They carry one of the bottom runs of the ladder, 50-50. They pass it on to their offspring. They might have a litter of 6, though. They might all get it, or they might all not get it, or you won't just get the 3 and 3, unfortunately. It's much more random than that. But over the thousands and thousands of cats in the world, it will be approximately 50. So, Tick Tabby is the top run of the ladder. And if you look at that yellow cat there, with the leg bars and the tail rings, okay, that is what Tick Tabbies all started like. And you still see quite strong leg bars in some ticked breeds, and you still see tail rings to some degree in some of the tick breeds. We have selectively bred out the Abyssinian, but the early pictures will show quite pronounced leg bars and tail rings, and often necklaces as well. And we have just selectively bred that out to get quite a clear coated. We'll probably never be able to breed out, not that we want to, that the M on the forehead, the pencil lines going from the eye to the ear, the dark eyeliner, the paler colored area, the spectacles, that is just a tabby term. Sorry, a tabby, that's just part of the tabby gene and the expression of tabby. They also have a ear liner. So all tabby cats will have a paler area just at the indent of the outer ear. So where the inner ear and the outer ear meet, you'll have a very fine sort of almost pencil line in the abbeys, I think, of the like a paler in that V of the ear, and that's the tabby gene at play. We also see very commonly the pale area around the chin, potentially lips, potentially nose as well. It shouldn't go past the chin onto the chest. If it does, it could well be that pesky Lockett gene. So we keep an eye for how far down that pale area goes. Perfectly normal on the chin. You'll see it in all tabby cats to some degree, and it's just part of what comes with the rest of the information on the T locust. So ticked is dominant. So if your Abyssinian gets out and mates with Tom from down the road, you would still expect a full litter of ticked kittens, even if he was something very different, because it's dominant. It would be 2-3 generations before you would see other patterns creeping in. So our cats are ticked. The breeds that are not ticked carry the non-ticked gene. The ticked gene is dominant, and it is one of the earliest expression of tabby. The others are naturally occurring recessive genes that crept in and were probably useful in the environment where the cat lived. So the mackerel tabby that was living in a forest or jungle or something, that would have been quite useful. It would have blended in better, it would have hunted better, produced more offspring, been more successful. So it would have gradually gained prominence in that area. Then the classic tabby is recessive to mackerel, and there isn't actually a spotted tabby gene. It's a probably polygenetic feature where the spots are just broken, and they're just very well broken. So it appears like a spotted cat, but it's actually a mackerel cat. The mackerel is just broken. The spots still follow the lines of what would have been the mackerel pattern. The exception to that is the Bengal, because they are a hybrid and they have a gene from that other group of cats. Remember at the start when I broke the cats down into the 3 subspecies, they're from the wild middle medium cats. So they do have a spotted tabby gene, and it's expressed in a horizontal flow rather than in the vertical flow of the broken mackerel. So I said at the start, all cats are black, all cats are tabby. So this is still true. All cats are genetic tabby. They are ticked, or they carry 2 copies of the non-ticked gene, and therefore they are then either mackerel or classic. So then I hear you say, well, what about Tompus, who's black from down the road? He doesn't have any tabby markings. He's just a black cat. So those cats carry 2 copies of a gene that suppresses the tabby pattern. They carry the non-agouti gene. At the top there, you've got the little a. If they've got 2 copies of the non-agouti gene, they'll be expressed as a non-tabby to some degree. The non-tabby pattern, this non-agouti gene, it is impervious over the red gene. So red and cream cats will generally still appear tabby, even if they're genetically not. And you often get some degree of ghost markings on some solid colored cats. And that's just the cat has two copies of the non-tabby gene, but it's only overrode expression of the tabby pattern to some point. And again, there's probably some other gene influence going on that's making that happen. So there we've gone from your black tabby cat to how we've got all the colors and patterns that we have. These are the main areas where I get quite a lot of my information from. Bessie Beast is a really good website. I wouldn't take everything on their word for word. She's not a geneticist, but she does have a very, very good working knowledge. And she's got a huge amount of information on there to cover all sorts of cat genetics right the way through. And once you've got a little bit of understanding and you know some of the terminology, it's quite easy to access her website and to find information and things that you want on there. She's got really good color and pattern charts that you can use to get from, how will I get some sorrel kittens out of these two usual adults? And the Book of the Cat 1980, that is a Bible. It's an all breed book, but it's got quite a good couple of pages on Abyssinians and Somalis. And then the foundation for all of my genetic knowledge really comes from Robertson's Genetics for Cat Breeders and Veterinarians. But there are not many cat breeders who could pick that book up and just dive straight in. You really need quite a comprehensive base before you can really get a huge amount from that book. And here we've got a lovely Abyssinian there, looking at us with a lovely tabby, ticked tabby pattern. Note the spectacles, the tabby M, the pale chin, all part. Can't see any pronounced necklaces there. We haven't got the right angle to see leg bars, but there's certainly none on the front. And it doesn't look like there'll be any tail rings either because we bred them out. But if you look just at the indent of the ear, you might be able to see it's a little bit of a pale liner just where the inner and outer ear. That is the residual part of the tabby. And then I put this black cat on, and you'll notice they have just a few white hairs there. So this again is that pesky locket gene that we haven't mapped yet, that we don't quite know what it is and why it's cropping up. So this was a breeding cat. It could very well have cats with full big lockets. It could very well throw that. We're not pretty sure it's a dominant gene, but the expression can be different from one generation to another. And the hair at the bottom is just to really demonstrate the ticking. If you look at the right corner of the screen, you can see that the dark tip, you've then got the nice bands of alternating color down the length of the hair. And when I was talking about the silvers, with the appearance of what looks like white at the bottom, if you do silver to silver to silver, that white will gradually creep up the hair, and you'll lose the ticking. You'll lose that. You lose it on the tip, the very tip last, but you'll lose the alternating bands of color if you're not careful and you don't go back to another color to help pull the ticking back and stop the inhibition caused by the silver. I'm going to stop talking now because my throat is like the Sahara. Shall we stop the recording?